I'm really excited to be here today and be able to share some of my research with the people that have been touched by the diseases. So in today's talk, what I'll talk about is treatment with a non-toxic, self-replicating antiprion. So in my talk, I'll briefly introduce prion replication. I'll talk about how we model prion disease in the laboratory. I'll talk about the existence of non-pathogenic prions and how non-pathogenic prions can be used as an antiprion to treat pathogenic prions. And then I'll summarize my data and give you the conclusions that we came to from this data, and I'll talk about some of my future plans. So for prion replication, I think it's really important that you understand the type of environment that prion replication occurs in. And so I've shown a picture of a neuron. A neuron is one of the main cell types that's affected by prion disease. And if you look at these cells, you can see all the extensions coming off the cell body. So these cells are built for communication, so they talk with other cells all throughout the central nervous system. And if you zoom in on the cell surface, it looks like a very busy city street. It has all these molecules that are attached to the surface, and they're in constant motion. And these molecules are proteins, and among these proteins is the cellular prion protein, so PRPC. Now PRPC is expressed by all individuals. It doesn't matter whether or not you have the disease, it's naturally expressed. When you have a problem is when it misfolds. And when it misfolds, it becomes known as PRP scrapie. And when you have PRP scrapie introduced to the brain, you have this very simple mathematical equation of PRPC plus PRP scrapie. So these two molecules will interact with one another, and PRP scrapie will convert PRPC to this disease-associated shape. And you end up with two molecules of PRP scrapie. And this is basically a cyclic process that's ongoing throughout prion infection. Now in the prion field, we're very lucky because we have some very nice mammalian models that fully recapitulate the prion disease. And these include hamsters, mice, and primates. And from what we've been shown from John Collingen's work with mouse models is that the infection process can be broken into two phases, an infectivity phase and a toxicity phase. So early on during infection, what you see is this rapid increase in prion titer. And what's surprising is when it levels off, you don't have this immediate transition to the toxic phase. You have to wait until enough toxic PRP scrapie accumulates in the brain, and then you move to this toxic phase. And when you move to this toxic phase, you also move to the symptomatic phase. So the animals, instead of being asymptomatic with no clinical signs at all, will become symptomatic, and you start to see a clinical phenotype that is completely dependent on the type of prions you've introduced the animals to. Now in this symptomatic phase is when the animals start to succumb to disease. And this is a survival curve that I'm showing in the bottom right. So you start to see this stepwise decrease in survival in the symptomatic phase, because when you subject a group of animals in the lab to the disease, they're succumb to disease at different times. So that's why you see this stepwise decrease. And this is the type of curve I'll show you throughout my presentation. So another aspect that's important to point out is in the toxic phase and symptomatic phase, you also have brain damage. This brain damage is also very specific for the type of prion disease you subject the animals to. And it's a direct consequence of the accumulation of toxic PRP scrapie. So you can see in this cartoon brain, you have accumulation of skull and crossbones. These skull and crossbones represent the accumulation of toxic PRP scrapie. And what's interesting is once you get to the toxic phase and symptomatic phase, this disease that lasts a long time all of a sudden becomes very rapid and very aggressive. So for this particular project, what we're hypothesizing are two things. First of all, we believe that non-pathogenic prions exist, prions that never cause disease, and prions that never cause toxicity. We also believe that these non-toxic, non-pathogenic prions, known as antiprions, or what we call antiprions, because we believe they can treat pathogenic prions. So since we want to use them as a therapeutic, I'll refer to them as antiprions. So we believe that once these antiprions are introduced to the brain, they replicate just like a pathogenic prion. They use PRPC as a substrate, and they convert it into another antiprion. And so when you have this accumulation, I've used smiley faces to represent the accumulation of the non-toxic, non-pathogenic antiprion. And I've used smiley faces because, as I just mentioned, you don't see the toxic phase, and you don't see the symptomatic phase. So when we're looking at candidates for the antiprion, what became very attractive were synthetic prions. So throughout the past 20 years, it's been a huge goal in the field to try to recapitulate the conversion of PRPC to PRP straight in a test tube and try to make an infectious product. And in doing this, there's been a lot of different methods that have been used to try to do this. And one common denominator is large-scale production of PRPC in bacteria. So basically the idea is to trick bacteria into producing PRPC of your choice, and then you can purify it. And this is much more efficient than purifying PRPC from mammalian tissue. So once you have this PRPC called recombinant PRPC, you can treat it two ways, either by chemical treatment or you can treat it by physical treatment or a combination of the two. So with the chemical treatment, you're treating with denaturants such as guanidine hydrochloride or urea, which just assists the protein in misfolding, or you can physically treat it by shaping or sonication, which are shown here. Now, there's a bunch of different recipes to misfold PRP, and once you have this misfolded PRP, they're in all different types of conformations that are dependent on the type of recipe you use. And you want to know whether or not you've actually created a synthetic prion. So what you have to do is you take these misfolded PRP forms and you subject them or introduce them into your laboratory animal. In this case, we're using hamsters. And the most common way to introduce the misfolded PRP to these animals is by directly injecting them into the brain. And when you do this, you wait and see if it causes disease in these animals, and then you know whether or not you've created an actual prion. And what's interesting is that very few of these recipes that have been used actually are highly pathogenic or highly infectious. Very few of them cause disease. Most of them don't do anything at all. And when we started taking a closer look at this, a lot of this work was done by Hippocampus. And what he showed is that some of these synthetic prions were actually replicating in the brain, but they're never causing disease, they're never causing toxicity, which is exactly what we're looking for an antiprion, and it also supports this hypothesis that non-pathogenic prions exist. So now you see this cartoon brain again with the accumulation of the smiley faces. So how do we think that an antiprion will work? What we think is that the brain has the capacity to support the replication of both pathogenic prions as well as non-pathogenic antiprions. So you can see in this cartoon brain you have both smiley faces and the skull and crossbones at the same time. So both of them are using PRPC as a substrate, and what we think is that there'll be a competition between the two, and it will reduce the amount of toxic prions, lower the brain damage, and delay the disease. And we hope they work efficiently enough so we don't have this toxic phase and we don't have this symptomatic phase. And I think a good analogy for this type of competition is just a simple race between somebody extremely fast, such as Usain Bolt, who's the fastest person in the world. He runs about 27 miles per hour, racing somebody that's slow, somebody like myself just walking, going 3 miles per hour. So when you look at this scenario, Usain Bolt represents the pathogenic prions, the guy walking represents the antiprions. And you look at this and you're like, well, Usain Bolt's going to win the race no matter what. But that's not necessarily true. If you give the guy walking enough of an advantage, he can actually beat Usain Bolt in the race. And that's exactly what we see in the lab setting. So what we're doing here is we're introducing the pathogenic prions into our laboratory animals. The animals we chose were hamsters. And when you do this, you know you have this finite period for optimal treatment. You want to treat before you have this rapid increase in prion titer. You also want to treat before clinical onset. So with this in mind, we set up 3 different types of treatment schemes. The first type of treatment scheme we did was post-exposure treatment. So this is more like the real-life situation. What we do is we introduce the hamsters to the pathogenic prions first, and then 63 days later, we introduce them to the non-pathogenic antiprions. So they're being injected with both pathogenic prions and the antiprion. And when we did this, unfortunately, we didn't see any change in the length of the disease. Both caused disease in 79 days. So we wanted to see if we could give the antiprion more of an advantage. So what we did is we injected the animals at the same time with the pathogenic prions and the antiprion. And when we did this, we do see a significant delay in the incubation period. So it goes from 79 days to 88 days. So even though it's just 9 days, it's pretty exciting because the type of disease that we're trying to treat is basically the Usain Bolt prion disease of hamsters. So it's very rapid and very aggressive. So we still wanted to see if we could make our antiprion work more efficiently. So what we did is a prophylactic treatment. Basically what we thought is we'd give our antiprion a head start. So we injected the animals in the brain with the antiprion 63 days before we injected them with the pathogenic prion. Now when we do this, we see a huge change in the disease. So the untreated group, shown in red, has an incubation time of 79 days. But when you give this pre-treatment or this head start for the antiprion, the incubation is extended to 129 days. So this is shown in green. So we're pretty excited about this because we're almost doubling the incubation period that we normally see in the hamsters, but we still weren't satisfied. We wanted it to work even more efficiently. So what we're trying to think of is another way to give the antiprion an advantage. So to give it more of an advantage, we found that, or we decided we would dilute the pathogenic prions. So in all other bioassays that I showed in the previous slide, we're using a dilution of 10 to the minus 2. So we diluted it to 10 to the minus 5, and we found that this dilution still caused disease in all of the animals that we injected. So we used this diluted pathogenic prion at a dilution of 10 to the minus 5, and we did the same type of pre-treatment with the antiprion. We injected with the antiprion 60 days before injecting with 10 to the minus 5 pathogenic prions. And what we found this time is that it completely prevented disease in 3 out of 5 animals. So for 600 days, you see no sign of disease in the hamsters. So it's basically prevented the disease. So by doing calculations and estimates based on our bioassays, we're able to find that 99% of prion infectivity was being reduced. So the last thing I want to show you is what the brains look like in the different groups of animals that we studied. So these are pictures of the hippocampus. This is the central region in the brain. It's a region that's known to be affected by this prion disease. And these slices have been stained for PRP scrapie. So PRP scrapie staining is shown by brown. So the more brown color you see, the more PRP scrapie you see. So in the healthy aged animals, you don't see any brown staining at all. So this means there's no PRP scrapie at all, which is what you would expect in a healthy individual. Now the groups that were treated with the antiprion alone, what we see is very mild PRP scrapie accumulation. You see the very mild brown staining, but you never see disease, you never see toxicity. So this is the smiley faces that have accumulated in the brain. So in the third group, the pathogenic prions, when you inoculate the animals with this, they succumb to disease, and you see all kinds of brown staining. So they have loads of PRP scrapie in their brain, and this is represented by the skull and crossbones in the cartoon brain. Now the group that stayed asymptomatic, the group that received the pre-treatment with the antiprion, never showed clinical disease. When we stain their brains, there's sort of an intermediate load of PRP scrapie. There's more PRP scrapie than what you see in animals that receive the antiprion alone, but there's much less than the animals that receive the pathogenic prion alone. So what this is telling us is that it inhibits the pathogenic prion from replicating, but it doesn't completely stop it, but it slows it down enough to prevent the disease in the lifespan of these animals. So just to summarize what I've shown, we developed a non-toxic, self-replicated antiprion that was generated from full-length recombinant PRPC. This is the protein that was produced in the bacteria. And when we inject this into the animals, there's mild PRP scrapie deposition, but there's no clinical signs. The second thing that I showed is that the antiprion significantly inhibited the pathogenic prions in the hamsters. I showed this both with the simultaneous treatment as well as the prophylactic treatment, and we showed that this efficiency was dependent on time and the pathogenic prion dose. And lastly, after the antiprion treatment, PRP scrapie accumulation was significantly lower and disease was occasionally prevented, and we estimated that prion infectivity was being reduced by 99% or by 2 logs. So the take-home message from this study is that prion replication can be separated from toxicity, and this provides us with a novel target for therapeutic intervention. So we have a lot of experiments ongoing in the lab right now, and they're based on these future plans. So what we're trying to do is to better define the mechanism for the antiprions, we want to know how they're actually competing with the pathogenic prions. The next thing we want to do is to determine whether antiprions can be applied to other prion diseases of other animals. And then lastly, we want to attempt to obtain more potent antiprions. One thing that would be very exciting is we could find an antiprion that replicates fast, but still never causes disease and never has toxicity. So with that, I'd like to acknowledge my mentor at UT Health in Houston, Dr. Claudio Soto. I'd also like to acknowledge Rodrigo Diaz, who's a former postdoc in the lab who did a lot of the studies that I presented here today. And then lastly, the CJD Foundation, once again, for their generous support, and the donors that specifically worked really hard to raise money to support my research. So thank you.
Charlie, we're going to do questions and answers. With the schedule change, do you still want to do 15 minutes, Debbie? Okay. So, any questions for the presenters? And I think we have roaming. Do we have roaming? Okay, it's up there.
Dr. Mays, I found your presentation very exciting as well as interesting. As you move forward, as you said, I mean, do you feel positive that this antiprion could lead to therapeutic possibilities?
Yes, I feel like it does have a lot of potential. I think we can learn more about the infection more than anything. As a therapeutic, you have to be concerned about certain things such as mutations. We know that prions have the ability to adapt and change by infection, so we'd want to make sure that it's very stable and that it wouldn't change and turn into something that's actually pathogenic. This is something that has been shown with some similar types of prions that don't show disease. If you keep on passaging them in mice time and time again, sometimes they will cause disease and sometimes they won't. So you have to really be careful with that.
I had a question for Dr. Mays too. So the animal model where you said there's no clinical signs, did the animal live its full lifespan?
Yes,
Okay.
It did. So the animal's natural lifespan is around 600 days. So more than 600 days, we see no clinical signs. And we don't see the neuronal death that you normally see when you inject them with the pathogenic prions.
I had two questions. One is, do other proteins misfold, or is it just the prion protein that is capable of misfolding?
Yeah, I mean, there's a lot of proteins that misfold. So something that's become very interesting here in the past 5 to 10 years is the prion-like mechanism of the proteins that have been associated with Alzheimer's disease, as well as those with Parkinson's disease and ALS. A lot of these have a protein that also misfolds and causes a similar type of cascade during their infection.
And just in my rudimentary review, I'm not a medical professional by any means, but I understand that there are proteins that help to come rescue misfolded proteins, like heat shock proteins and other proteins. Have there been any studies where, as opposed to using, drug agents, but to use other proteins to assist in curing this disease or preventing this disease?
Yeah, I mean, what you're referring to with the heat shock proteins is based on what's been found with yeast prions. And that's very interesting. That's not been looked at really in depth with mammalian prions. There's been some work done where you can see some things with some in vitro assays, but not a whole lot in the animal assays.
Yeah, I think that there is some data on transgenic mice overexpressing some HSP, but there is no so much difference in the incubation period and so on.
And I think your other question about other protein misfolding disorders, I think Dr. Cashman's going to talk a little bit about that in his talk coming up. So. There's someone down here. I was wondering if I could ask you a question, Stefan. We hear a lot about translational medicine, translational research, And obviously, the families get really excited when they hear about potential treatment targets. But could you talk a little bit about what translational research is and why that's important when trying to look at treatments?
Okay, generally?
Yes.
Yeah. Yeah, okay. From the Prion researcher point of view, in the studies that we perform on clinical trials in CJD, what was very frustrated, in fact, it was that the compounds that we tried to test where the scientific basis leading to their use in humans was very low, in fact. For example, for quinaquine, in 2002, it was only results using a neuroblastoma murine mouse cells treated, infected with a scrappy strain. And on this basis, we go on to clinical use. And it was also not so much the case, but a little bit with doxycycline, because we go to clinical trials on the basis on in vitro studies and some in vivo data, but only in hamster chronic infected using scrapie. So I think that now we have all the models that can be used to have to be more convinced by the potential of the drugs by using genetic models with human PRP expression, by using cells infected with human prion isolates, and of course transgenic mice infected with prion strains, because it is very important to prove the effect on human strains. So, of course, it depends on the mechanism used for therapeutics. If you simply inhibit the expression of PRP, so you can expect that whatever the strains, you will be efficient. But for drugs that are targeting the PRP conversion of the PRP aggregation or accumulation, it could be that it is conformational specific. So you need to demonstrate that it can be efficient against assemblies of human PRP involved in natural disease.
Dr. Mays, where do the antiprions that you inject come from? Are they a component in the blood, or where do they come from? Where do you get them?
So that's what I was explaining with the bacteria. So we basically are tricking the bacteria. Bacteria naturally don't express the prion protein, and so we introduce the DNA that encodes for the prion protein, and then we purify it from the bacteria. So basically that's why we're referring to it as being synthetic. So it's basically an artificial protein that's been misfolded.
What other animal models would you anticipate using before we get to humans?
So yeah, the question was what other animal models would we use before we get to human? Yeah, what we're looking at now are transgenic mice. Transgenic means they basically have altered DNA so they can better represent other types of prion diseases. So we use transgenic mice that express human PRPC and see if it works against the human disease. That's the most straightforward next step.
I have questions for each of you. Stefan, welcome. How is it that the E200K worms, the C. elegans, are expressing a misfolded protein when they live for such a short span? Why is the protein toxic? Why is it PRP scrapie resistant? Why is it misfolding? And then for Charles, my question would be, given the fact that these antiprions can cause animals to die, how are you going to avoid that? How are you going to be certain that you're not causing harm?
Go ahead, Steve.
Okay. Thank you for your question. Why? I think that the most important thing is that the prion protein is overexpressed in these neurons, and naturally that they do not express prion protein. So it's a gain of function, and maybe because it is overexpressed, in 3 days we observe no toxicity. And what I have to say is also that other neurotoxic proteins that are overexpressed in mechanosenstive neurons or other neurons in this model can be also neurotoxic. So an important point is that we detect protein ASCA-resistant PRP, meaning that it seems that the assemblies or the conformation of human mutant PRP in these animals is pathological. So why? I cannot say why, but it's an observation, and it is not observed in animals that are expressing the wild-type PRP. So the hypothesis is that the mutation gives to the protein a proportion to misfold.
Yeah, Dr. Gershwin, so you ask, how do we make sure that they don't mutate and cause disease? That's one thing I tried to address in the first question is basically what we've done is we've followed the protocol that was described by Ilya Baskakov. We've made some minor changes, and we've injected about 15 hamsters and never saw disease. We saw it accumulate in all of them. And what we're doing next is doing a serial passage to see what happens, to see what type of adaptation there is. We know that with some of the methods that he used, you do see an adaptation just like you would see jumping the species barrier, and it does cause disease, but it's still very slow. And then in some cases, you still don't see a disease, but you do see it replicating in the brain for multiple passages. So sometimes, in some cases, with very specific protocols, it may never cause a mutation. But yeah, that's something you want to be very confident about and make sure it doesn't mutate and change.
Is this working? Hello? Is there any indication that these antiprions are in one single conformation? Do we have any structural information on them? Is it a blend of conformations? Do we know any of that?
Yeah, that's one drawback in the field. I mean, that'll be a huge breakthrough when we're able to figure out the structure of PRP scrapie. That's one of the major goals, I think, for the field and really the next big finding, because that will also help us with treatments in general, because then we'll be able to know how to treat them if we know exactly how they're folded. Because what we believe from what we see is that each disease, that the PRP scrapie is in a different type of conformation.
My question was for Dr. Hike. Hey, Kay. I'm not sure. Stefan. There were 17 sort of drugs that you said came out. Will you be testing with them? Is that what your thought is? Like you had an antihistamine, you had antidepressant, you had anti-inflammatory. Is that something that you're hoping to do in an animal model?
Okay. Your question is about the diversity of the molecular compounds. We observed some compounds that were expected, in fact, such as anti-inflammatory, non-steroid anti-inflammatory drugs, because there are some publications in the literature showing that these drugs can inhibit the neuronal death due to PRP-SC or PRP peptides. So it was not surprising to detect these compounds. We have also detected some compounds that were previously identified as antiprion compounds, such as phenothiazine derivatives. So it is reassuring for us. It's like positive controls. We are able to detect drugs that have been previously described, but also some new drugs. So they are from different classes. Some are dopaminergic, the other are anti-dopaminergic, anti-serotoninergic, pro-serotoninergic. Thus, maybe it is not their effect on prion disease. Maybe they have an effect on PRP conversion of accumulation or binding to PRP. So we have to explore that next.
Okay, one more question.
Hi.
One time.
My question is for Charles. So have you tested the recombinant, the normal form of the PRP, like without the misfolding? The reason I ask this question is because we previously also showed in vitro, and the recombinant PRP can inhibit the conversion of the PRP.
Right. So what Wintron's referring to is in PMCA, or protein misfolding cyclic amplification, you can add recombinant protein that hasn't been misfolded. It's in the normal healthy conformation into the assay, and it basically blocks the conversion of PRPC into PRP scraping. So that's what he's referring to. We've shown similar things with our PMCA situation. It seems like that when we add our antiprion into PMCA, that it does block the conversion. That is one important control that we didn't look at, is we didn't add in just the recombinant hamster PRP, and I think that'd be very interesting to look back at.
So please join me in thanking our grant recipients and speakers. Good job, Will.